usually the lectures are in English.
Oh, last semester
All the lectures were in English.
Ouais, actuellement,
That's everybody else.
donné en anglais. Pourquoi tu l'as préparé en français, au complet?
C'est un cours de l'Université de Montréal. On parle français, on enseigne en français. On a pas fait de demande spéciale. Mais moi, je peux donner en anglais, je parle anglais. Ça me dérange pas.
Absolument, c'est en anglais pour compter les personnes qui sont bilingues anglais. Je suis encore avec toi. On peut faire un sondage au début.
Oh oui, moi, ça me dérange pas. On va faire
No, it's good. You don't know.
I don't really.
Okay. Hey, it's supposed to, all the courses in University of Montreal are supposed to be French.
I know.
But I don't care if you practice in English. Yeah.
Alors, bonjour tout le monde, je m'appelle Audrey Klein. Il y a eu une demande de donner de cours en anglais, donc je me demandais si ça convenait à tout le monde. Everybody speaks English? Yes? Okay, we'll do it in English then.
Thank you.
Today I was invited to talk to you about deep-rooting coupled receptors. So, how many of you are familiar with these proteins? 1, 2, 3. Do you guys actually have research projects using this stuff? Okay. The professor responsible for this course has an announcement, so we're going to respond.
Alors, bonjour tout le monde. Bienvenue au BIM 6028. Il faut juste prendre 2 mots avant la conférence d'aujourd'hui, juste pour si vous avez des questions en fait, parce que je pense que vous avez reçu les synopsis. Donc vous savez un peu comment ça va fonctionner. Il y a 2 examens. Il y a un intra et un final. Okay. Vous avez probablement les dates, mais pour vous les donner. L'intra, c'est le 25 février. Et c'est à l'heure du cours, donc ça va être à 16 heures. Le local, ça va être à déterminer. S'il n'y a pas d'autre local, on va le faire ici, mais je sais que si c'est pas le plus pratique pour les examens, c'est que si je trouve un local où il y a plus d'espace pour travailler, on va se déplacer. Et le final, ce sera le 14 avril. Même chose, à l'heure du cours, le local en verra. Et donc l'intra porte sur les cours qui ont lieu avant cette date-là, donc du cours d'aujourd'hui jusqu'au cours du 18 février par Will Sack. Et le final porte seulement sur la deuxième partie, donc les cours d'après. Donc le final ne couvre pas l'ensemble des cours. Okay? Donc l'intra pour la première partie et le final pour la deuxième partie. C'est clair. Chaque année, il y a toujours un peu d'ambiguïté sur qu'est-ce qu'il faut faire dans les examens. Là. Donc l'intra, c'est pour la première partie, puis le final pour la deuxième partie. Ça marche?
Sorry, just so I understood correct, half of the course is in the midterm and the rest is in the second?
There are 2 exams.
Yes, the midterm is...
And one midterm, which is 25th of February.
Right.
And this will cover all the classes, the lectures from now until then.
Yes.
And the final exam in April will cover all the classes starting after the intro.
Right.
Okay? So there's no exam will cover the whole class. All right. Ces 2 cours, ils ont chacun un cycle différent. Celui-ci, c'est 60-28. J'ai toujours de la misère à l'étonner. Puis l'autre d'avant, je pense que c'est 70-26. Non, c'est 2 cours séparés. Vous allez normalement avoir une note séparée pour chaque cours.
C'est 6 crédits ce cours, mais 3 crédits.
cours quasi chaque. Parce que je pense que dans certains programmes, certains étudiants peuvent prendre juste un ou l'autre, et dans d'autres programmes, ils peuvent prendre les deux, mais c'est vraiment deux cours qui sont intimement liés, mais c'est deux cours avec deux notes. D'autres questions? Pas de questions.
So my name's up there with my email address, so if you have any questions before the exam, you didn't understand a concept or something, you can just send me an email, you can take an appointment, discuss further, or maybe come back. No problem with that. I'm at the University of Montreal at the Department of Pharmacology. So today what I'm going to talk about is first the structure and function of G protein-coupled receptors. So as you may know, these are a very important class of membrane proteins. So we're going to talk about their structure and after that their function. All the different signaling pathways they may engage. So obviously their name is G protein-coupled receptors. So their main effectors are G protein, but they can transduce signal independently of G protein as well. So we're going to talk about that. After that, we're going to talk about the mechanism that leads to their desensitization. So desensitization is a mean by which receptor can control their own response by regulating either the intensity or duration of response. After that, we're going to talk about the mechanism of internalization, intracellular trafficking. So once they're desensitized at the plasma membrane, they will get inside the cell in order to be dephosphorylated and then recycled back to the plasma membrane or sent to the lysosome. And at the end, we're going to talk about more current concepts. The first one will be bias signaling. So different ligands or drugs can bind the receptor and engage specifically certain signaling cascades. So we're going to talk about that. And after that, a good example of this is activation of the NOT kinase pathway. And we're going to see and discuss the different signaling cascades that are activated by G protein-coupled receptors to lead to MAP kinase activation, which is important in the context of proliferation. So why are these membrane proteins interesting? Well, for many reasons. First of all, they're very important pharmaceutical targets. So today we estimate that about 40% of the drugs that are on the market target these families of membrane proteins. Following sequencing of the human genome, it was estimated that about 1,000 genes encoded G protein-coupled receptors. So 600 are mainly present in the olfactory system. So this is the way you can sense and detect different odors. 200 are very well characterized, so you can think about beta-adrenergic receptor, angiotensin receptor, serotonin, dopamine receptors. We know them well. We know the ligands. We have drugs that target these receptors, and about 200 other are less characterized. So they're either still orphan. We don't know which signaling pathways they engage. So there's still space to do research in this field. You may know that activation of GPCR leads to numerous amounts of physiological responses. You can think about contraction of muscle, vessel tone, neurotransmission. So they're everywhere, and this is why they are so important in physiology. So to acknowledge their importance, the Nobel Prize of Chemistry was given in 2012 to Bob Lefkowitz and Brian Kobilka, a former postdoc of Bob. So Bob actually discovered the beta-2 adrenergic receptor, and following identification of this receptor, well, others could identify other receptors, and this has become a large family of membrane proteins. And Brian was the first one who was able to crystallize these proteins. So obviously it's easy to crystallize soluble proteins, but membrane-bound protein is another story. So different methods to stabilize the structure. He was able to crystallize them, and this is important for drug design once again. So in the next slide, I present you a different type of cartoon. It's kind of an oversimplification. So I just want to point out here that these membrane-bound receptors can be activated by a great variety of ligands. These vary from light to calcium and others, like I mentioned, but also small molecules and proteins. So you may recognize here different peptidic hormones that you know well, nucleoside, and glycoprotein as well. So all these ligands have the same ability to bind the receptor at different places, but then engage a change in conformation. And it's this conformational change that will allow the activation of the G protein, and the G protein in turn will activate different types of effectors, which can be either present in the membrane or in the cytosol, and together they will engage intracellular signaling to produce intracellular membrane. So before we go into the signaling cascade engaged by the protein coupled receptor, I just want to talk to you a bit about the different classes of the protein coupled receptor. Because if you're interested in the 200 that are not well characterized, what is the first thing you're going to do is to look at the sequence and then try to classify them. So what kind of receptor is this? Is this a rhodopsin-like kind of receptor? Rhodopsin receptor were the first to be identified, and they've become a prototypical model for this class of membrane proteins. So family 1 is all rhodopsin-like receptors. 1A, 1B, 1C. They're differentiated by the binding site for the ligand, but this cannot be detected in the structure. What can be detected in the structure is the different motifs that are present within these subtypes of GPCR. So first, there's a dry motif in the second intracellular loop. So maybe it's not clear on the image here, but I think you can see it well in the slides, and I'll come back to that when we talk about spontaneous activity. So the dry motif is very important for G protein coupling. So these receptors clearly have this motif. Also, they have a cysteine bridge between the loop, the extracellular loop 1 and 2. They also have an NP-XXY motif in the 7th transmembrane domain. Furthermore, they have a pantosylation site at the beginning of the C-terminal tail of the receptor. And when you look at the crystal structure of this receptor, there's an anti-parallel alpha helix that is within the first part of the C-tail here, because it's hooked up by the palmitic acid. So either 1A, 1B, or 1C all have these features, these common features. The second family is the one of the gastrointestinal peptide family. So the prototypical is the secretin receptors. So although they also have 7 potential transmembrane domains, they don't have the specific motifs that I just named here. So there's less receptors that belong to this family, and as you can see, they're all from the gut. The third family is characterized by a very large extracellular domain, which resembles a Venus flytrap. So this large domain will bind the ligand, and again, upon binding of ligand, will induce a conformational change within the transmembrane domain that will lead to activation of the receptor. And the prototypical receptor for family 3 is really the glutamate receptor. Glutamate being a excitatory neurotransmitter. There's also family 4 and 5, but we are not going to talk about them today. So 4 is for pheromone receptor, and 5 is for the atypical GPCR, which are the frisal and the Susan type of receptors. They function a bit differently. So just focus on the 3 first family that I've mentioned, with rhodopsin being the largest. So if we want to talk about engaging signalization, we can think about binding of ligand and, again, like I mentioned, change in conformation of the receptor and then activation of specific effector. But there were different reports a long time ago that mentioned that some pathologies were caused by actual activating mutation of receptor, and these receptors no longer needed ligand to be active, and this obviously caused a problem. So a few examples are highlighted here. First of all, it's a report describing constitutive activity of the rhodopsin receptor, which leads to blindness, so it's a major problem, not being able to control activation of this receptor. There was also somatic mutation reported in the serotropin receptor gene, which caused hyperfunction of the thyroid and led to adenoma. So again, an important role in pathophysiology. And another example illustrated here is a mutation of the LH receptor associated with early puberty. So all these receptors actually are mutated in specific residues that allow spontaneous cooking to G protein. So I was describing this dry motif in the second intracellular loop here. And you can see that if we decide in the lab to play with a well-known receptor, like the beta-2 adrenergic receptor, and mutate all the different amino acids, we can find activating mutations. So one example is the mutation of this dry motif here. First of all, the D residue. And if we measure activation of the cyclic AMP pathway, this receptor is coupled to GF, and we are going to talk about the G protein in a few slides. But let's look at this measure of, if I can say, activation here. And this is in the condition where receptors are transfected in cells using different amounts of DNA. So the further you express a receptor in cells, the more chance it has to come randomly in contact with the G protein and activate the adenylyl cyclase and produce cyclic AMP. But this is nothing compared to addition of the ligand. So all the different curves here illustrated by the wild type just show that the more you overexpress a receptor, the more basal activity you may have. If we mutate this D residue here, you can see that suddenly the transfection of a few receptors leads to a big response in cyclic AMP. And this is the same if we change the residue for alanine. And here there's another example where these residues were mutated, so constitutively active mutant, and again, no ligands added, just transfection of this mutant leads to activation of cyclic, generation of cyclic AMP. In physiology, only one receptor is always high at basal, and this is the ghrelin receptor. So about 50% of the maximal response is the basal response, okay? So when you treat with the agonist, of course, you can further enhance activity of this receptor, but already at basal level, it's really high. And why is it high? Because this receptor is associated with anxiety. So unless you would have high basal, you would never eat, okay? So because we need to grow and have an active metabolism, this receptor is high. So if we look at the profile of inositol phosphate accumulation, which is a readout for GQ activation, you can see that at basal we already have a very important response. We can lower this response with an inverse agonist, and we're going to discuss that later. But when we add ghrelin, we can further enhance this response. Same thing if you look at activation of the G protein. Ghrelin obviously increases activation of the G protein, but at basal level or at resting level, the activation of the G protein is already very high because the conformation of the receptor is, let's say, half turned on nature. And people who have mutation in this receptor will be very small stature, and they will have major problems in gaining weight. So if we come back to the G protein, I think you've heard of these before, so I'm just going to summarize quickly the different signaling pathways that can be activated by GPCR through these G proteins. Just here a first cartoon to show you the receptor present within the membrane, and then the G protein associated on the intracellular surface. So usually when we draw them, we draw big receptor and just little G protein, but in fact when people actually crystallize the complex, they were able to show that the G protein occupied a great part of the receptor and the complex of the heterotrimeric G protein. So the alpha, beta, and gamma subunits are quite substantial. So you can characterize receptor according to the G protein they activate also. We'll talk about GQ coupled receptor, GF, GI, or G1213. So for the receptors coupled to alpha F, F is for stimulation of adenylyl cyclase, and the example are the beta-2 adrenergic receptor and the vasopressin receptor also. These G protein will lead to the activation of adenylyl cyclase, which will in turn convert ATP for cyclic AMP. Cyclic AMP is a major second messenger himself. So cyclic AMP will result in the activation of protein kinase A, and like any other protein kinase, they're very important to phosphorylate substrates themselves to engage signaling and cellular response. So the second class is the GQ coupled receptor. G11 is an homologous form of GQ. Many receptors are coupled to GQ. You may know the angiotensin, the endothelin, oxytocin, bradykinin receptors. So these receptors will, through GQ, activate phospholipase B. So the phospholipase will obviously transfer membrane lipid into diacylglycerol as well as inositol phosphate. Diacylglycerol will directly activate protein kinase. IP3 will go bind its receptor on inside the cell and promote liberation of calcium from intracellular protein. After that, we have GS. We also have GI, so receptors that will inhibit adenylyl cyclase. And there's a few examples of that. You may know CXCR4 receptor, which are important for motility, FAT receptors, so the platelet activating receptor. So they all couple to GI. They all block activation of adenylyl cyclase. As a result, we can lower cyclic AMP. So a good way to assess activity of these receptors is actually to biochemically stimulate adenylyl cyclase using forskolin, which will bind directly the enzyme and activate it. So we have increase of cyclic AMP and stuff. And then you just throw an agonist and you'll see cyclic AMP completely go down. And finally, there's G1213. So this heterotrimeric G protein is linked to activation of Rho GPase. Rho is important for cytoskeletal remodeling and to the regulation of phosphorylation of myosin and cells. So we'll detail using cartoon these different pathways in it. So what I'd like to show you is the diversity of G proteins. So I just aligned them by drawing a cartoon that is quite simple, but in fact, there's not just one GS protein. Depending on the nature of the cell or the tissue, different isoforms can be expressed. So for S, we have several isoforms. Same thing for I, Q, and 12 as well. And they can be activated or inhibited using different toxins. So pertussis toxin is a good inhibitor of GI. And they can be linked also to different subunits to different effectors inside. Same thing for the beta-gamma subunit. So there are many gamma and many beta subunits. So you can imagine in a neuron the complexity of the G protein that can be completely different from the same GI protein expressed, let's say, in cardiomyocytes. So the different combination just tweaks a bit the function of the G protein and dictates the different pathway it can activate. So we don't know exactly the different function of these beta-gamma subunits. Maybe in one concept, the gamma-5 is very important for such and such effect, but these are being figured out right now with novel techniques that have been developed where we can do different combinations of these quite rapidly in assess cellular response. So we go back to GS. I've used cartoons to illustrate the different signaling pathways that I presented rapidly on the different sites. So let's go back to the receptor here present at the plasma membrane. So upon ligand activation, it can couple to the GS protein. And you can see in the cartoon here that they are drawn separately. So before we would say that activation of the G protein led to dissociation of the alpha and beta-gamma subunits. Now we don't think it's so true. We think it's just a tridimensional reorganization of the three subunits that open a new interface for interaction with the different effectors. So the alpha S subunit is actually the subunit that will go and bind the adenylyl cyclase. And again, there are many adenylyl cyclases within the cell, which will in turn convert ATP to cyclic AMP. So cyclic AMP, like I mentioned, is able to activate protein kinase A. And here it's illustrated as the four different subunits that form this pKa. So pKa consists of two regulatory subunits and two catalytic subunits. So two subunits that contain the activity, and the regulatory subunit can bind, as illustrated here, cyclic AMP. So the regulatory are in black, and the active subunits are in blue. So pKa, upon binding of cyclic AMP, will change its conformation, obviously, and go phosphorylate different targets, which can be associated with vasorelaxation, gene regulation, channel regulation, and growth. So many effectors downstream of pKa. And I'm going quickly over this because I think it's you've seen this. So here I've highlighted in red the different major pathway activated by GQ. So we still have our receptor at the plasma membrane. Upon binding of the ligand, we have activation of GQ, so binding of GTP on the alpha subunit. And the main pathway activated then is activation of phospholipase C, like I mentioned. But other signaling events are also activated directly by GQ, but I will not discuss them in length in this session. So activation of PLC promotes transformation of PIP2, so phosphatidylinositol bisphosphate, into diacylglycerol and IP3. And like I mentioned, diacylglycerol will go and activate PKC, an homologue of pKa, although the structure is not the same, which will go and phosphorylate its different effectors. In turn, IP3 will bind its receptor, and promote release of calcium from the intracellular source. So I do not want you to learn this by heart or go in detail. It's just the main pathways that are important to memorize. Of course, if at 6 o'clock you have not understood that GQ leads calcium generation or calcium accumulation itself, well, maybe I've not done my job properly. Maybe because it's in English, I don't know, but this is the take-home message. I won't go to GI because GI is like GS, but it's blood cyclic AMP activation. I'll just present you the G12-13 pathway here, where RHO-A is absolutely central to activation of this G protein. So you can see G12-13 here and here. They both lead to activation of RHO-A. And RHO-A in turn can activate different signaling pathways, which are linked to numerous responses associated with actin remodeling. Okay, so I was talking earlier about the fact that GPCR can transduce signal without activating G protein. So I'm going to give you two examples. And this is important to remember because these proteins do not exclusively bind to protein, okay? They can transmit signal via other So the first example is the beta-2 adrenergic receptor that can interact directly with a protein named NERF. NERF is for sodium proton exchange regulatory factor. So this NERF protein obviously dimerizes and binds NHE3. So NHE are the sodium proton exchangers. And activation of these pumps is actually important for resorption of sodium in the kidney. Okay. So physiologically, this signaling pathway is very important. If we mutate one of the amino acids that is responsible for the direct interaction of the carboxy terminal tail of the receptor with NERF, we significantly impact sodium absorption. So it's really the last 4 amino acids of the tail that are responsible for binding of the beta-2 to NERF. So not all GPCR ends opposed to NERF, obviously. This is unique, almost unique to the beta-2 receptor. And, many people do molecular biology in here, and we're using molecular biology to tag protein at the C-terminus. So people had made GFP beta-2 receptor because it's super easy to follow using different microscopy technique and see it move around. But addition of a GFP at the C-terminus completely blocks this interaction because it's a big 20-something kilodalton protein that you just hook here and you prevent this interaction. So these receptors are effective when put in renal cells to modulate absorption of sodium. So be careful when you tag protein either in the N or C terminus. You can prevent such signaling events. The other example that is well characterized is activation or binding of the Homer protein directly to the mGluR receptor. So these family tree receptors can directly interact with these Homer proteins. That's also dimerize and link the receptor directly to the IP3 or the adenine receptor. So activation of these two receptors will promote calcium release from intracellular space. So this is a way of bypassing, if I can say, GQ activation, which activates LC, generate IP3 from CYP2, and then IP3 go bind their receptor. For this receptor, it's found a way to bypass the whole G protein signaling and directly bind an intermediate effector to activate the IP3 receptor. So there are some G protein independent signaling events also. So in the next section, I'd like to talk a bit about the mechanism responsible for receptor desensitization. So the desensitization, you can practically experience it every single day of your life. You enter a room, it smells super nice, and then maybe 30 seconds later, you don't have that smell again in your head. So it's because your receptor has been sensitized to that organ. So if we do a classical pharmacology and we just paint, let's say, an artery in an organ back and stimulate it with vasoactive contractile agent, you can say that upon the first stimulation, you'll have a very nice contraction of the vessel. So the receptors are fully competent. They respond maximally to this agonist called A. Okay? Then you wash out the agonist, you let the tissue go back to its initial tension, and you stimulate again, let's say an hour later, with the same agonist. And then you don't get maximal response, you only get a partial response. And finally, you wash out the agonist, let it rest, and then the third response is absolutely nothing. Okay? So this illustrates well the fact that this tissue has desensitized to the agonist. It can no longer respond to that agonist. And this is a cartoon illustrating maybe contraction of the vessel, but you can also measure activation of adenylyl cyclase through generation of cyclic AMP, and you'll see upon repetitive stimulation that you never get the maximal response. So how is that possible? Well, it's important for GPCR to control their own responsiveness, okay? Because some signaling events may be not life-threatening, but life-saving. Let's say you're hiking in the woods and you're having fun, and then you see a bear. Okay, you climb up, bear's climbing, and you escape. So you're very afraid. You have a big adrenaline rush, and then you climb down and you continue your hike, and the cub comes running after you. Well, you better have enough adrenaline to push you back up because your life is in danger. So for adrenaline, receptor recycle and resensitize very rapidly. Okay, so we don't get much desensitization because of the so fast resensitization process. But this is uncommon. Most receptors, they stay desensitized for a long period of time. So my point was that desensitization and resensitization of the receptor can be very important in physiology. So the mechanism that will control these events, they're very precise, okay, and have become very well characterized over the years. So the first event that occurs upon sustained activation of a receptor by an agonist is activation of enzymes that will serve to phosphorylate serine and threonine residues present on the intracellular part of the receptor. So these kinases, either the GRK, which are specialized to phosphorylate GPCR, so G protein coupled receptor kinases, phosphorylate the receptors, but also the protein kinase that you've activated once the receptor coupled to the G protein. So PKA and PKC, they can also come back to the receptor and phosphorylate the receptor. So the first event is really phosphorylation of the receptor by either second messenger kinases or G protein coupled receptor kinases. And are the function of these two families of protein different? Yes, they are. They can both phosphorylate serine and threonine residues, but in one case, GRKs are very specific and phosphorylate only receptors that have seen agonists, so that have been desensitized or bound to the agonist. While protein kinase of second messenger, they can either phosphorylate a receptor that has seen an agonist or one that has not seen an agonist. one example of this heterologous desensitization is, let's say, the context where you would stimulate cells with adrenaline, so you would generate cyclic AMP in cells, like say PKA. The PKA would come back and phosphorylate the receptor, but at the same time, the PKA would go and phosphorylate, let's say, vasopressin receptor. Okay? So if you challenge the cell with vasopressin, even if it's for the first time, because you pre-stimulated them with adrenaline, they will not respond maximally. Okay? So we get heterologous desensitization. Even though a receptor has never seen an agonist, it will be desensitized through this mechanism. While GRK will not do that. It will only phosphorylate receptors that have seen the agonist. And we can measure phosphorylation of the receptors experimentally. So if you're not afraid of hot phosphate and you are willing to go in the hot room and label yourself, well, you can put 32-phosphate on the receptors, bring these back in the lab, IP the receptor, and just run it on a jet. And in the control condition, you will have not a lot of phosphorylation if you wash well your cells, but in the desensitized condition, so in the condition that where the cell will have been stimulated with an agonist, you will see a lot of incorporation of hot phosphate in your eye. So you can see this experimentally. So I'm going to detail more the GRKs because they're so specific and so specialized for phosphorylation of the receptor that they're important in what we're discussing now. So there are serine threonine kinases. You may know tyrosine kinase receptors, so receptors like VEGF receptor, they have tyrosine that becomes phosphorylated upon signaling. GPCR, it's not tyrosine, it's serine and threonine. So the enzyme will phosphorylate these two types of proteins. 7 isoforms of GRKs have been identified. GRK1 and 7 are only present in the visual system and will phosphorylate the rhodopsin receptor. GRK2 and 3 constitute the second class. So first of all, they were named BARC1 and BARC2. So BARC1, GRK2 was really the first identified. Sometimes if you do literature, some people will still use the term BARC. BARC is for beta-adrenergic receptor kinase. So it was the first enzyme isolated out of the study of sensitization of the beta-2 receptor. But after 7 more clones, we changed it. So these two isoforms are mostly present in leukocytes, the cortex, the heart, the lung, and the kidney. And then a fourth class, which is either distributed in very specific area or their function is a bit different from the classical GRK, are GRK4, 5, and 6. This is schematic representation of the GRK. The 6 of the 7 isoforms are drawn here. I'd like to bring your attention to the catalytic domain that is flanked right in the middle of the protein. In the N-terminal region, we have the recognition site for the receptors, so it's important for them to be able to recognize the activation receptor. So the features are within the N-terminal region. And in the C-terminal cell, we have binding sites for different partners. So one of the key partners of the GRK is the beta-gamma subunit of the G protein. So it's very convenient. Upon agonist stimulation, you have activation of the G protein, and the beta-gamma subunit serves as an entering site for the GRK. Other binding partners have been identified also. HIP2, so membrane lipids stabilize the recruitment of the kinase to the plasma membrane. PI2 kinase is also a partner of the GRK. component of the actin cytoskeleton and other proteins. The second step in the desensitization process is really the recruitment of the beta-arrestin protein. So arrestin was named arrestin because it arrests G protein signaling. So phosphorylation of the receptor does not assure desensitization. It only serves to create a high affinity binding site for these beta-arrestin proteins. So beta-arrestin proteins will come and bind strongly and efficiently to the activated receptor and physically prevent coupling to G protein because it's so big and it doesn't dissociate. So there's no way for G protein to come and bind again the receptor. So it really secure desensitization and no further response to agonists. So assures uncoupling of the G protein. So we started with 1,000 receptors, 7 GRK. Now we're left